Hi, everybody, and welcome to the Genetics Podcast. I'm really excited to be here today with Dr. Mark Kotter, the CEO and founder of bit.bio, a company that's developed what I think is an amazing and novel method for cell reprogramming. Mark has a very compelling vision for building a platform and a company that can program ultimately every human cell type. We're not there yet today, but we're going to talk about what it might take to get there. And this, of course, has applications in fundamental research into basic biology and the building blocks of life, as well as medical applications like cell therapies. So we're going to dig into each of these hopefully today. And Mark, first of all, thank you so much and welcome to the podcast.
Patrick, thank you for having me here. Really good to be on the podcast.
I'd love to start if you could take us back to when you were actually first introduced to the concept of stem cells and cellular reprogramming. You were not a scientist initially, you're actually a surgeon, and I'm really interested to hear what got you so interested in this topic and caused you to maybe change career trajectories from what you might have imagined when you started medical school.
It's been quite a journey, quite frankly. I started off actually studying maths and physics, but halfway through the course felt that this was not taking me into the future that I desired. So I sort of had a deep crisis, rethought what I needed to do and what my purpose was in life, and made the choice to switch tack and go into medical school. And in medical school, I very quickly realized I was pretty drawn into the why questions, so the research in addition to the application. And so I was very lucky that during that time I met my later PhD supervisor at the University of Cambridge. And the idea that I had was to somehow combine something to do with the brain and the spinal cord with my future career. And so after I finished medical school, I joined Robin Franklin's lab at the University of Cambridge, and we were investigating regenerative processes in the brain and spinal cord. And we didn't actually know that we were studying stem cells, essentially. That wasn't a particular topic at that point in time. But very rapidly, of course, it became a topic. And through a lot of luck and hard work, I ended up a few years later, a decade nearly later, as a PI at the University of Cambridge. And I was part of what were the early beginnings of the Stem Cell Institute under the umbrella of Roger Peterson, who was one of the founders of embryonic stem cell research. So that put me in a very, very, very important spot where I was able to work as a surgeon, already sort of undergoing my training, but at the same time, I was able to do some research and continue my own research. And one of the things that I noticed was that there's a big difference between human and animal biology. Just by studying the same cell in animals, models, but also then from surgical samples. But the whole reason why I went into neurosurgery, was because I had this idea of using cells to promote repair.
And where was the field at when you started your PhD, and where is it at today? What have been the major changes? And then what are the things that maybe stubbornly haven't changed as much as we might like?
There was some really radical changes since then. Probably the biggest was initially that wave of realization that human stem cells could potentially provide a source of any cell type if we could somehow harness them. Turns out it's much more difficult. And although we've been working with human stem cells for nearly 30 years now, there's still no product in the clinic. There's no treatment at the moment. And that just tells you how difficult it is to control these. That was a wave of interest that came together, and that was the reason why the Stem Cell Institute in Cambridge was formed. But in terms of scientific breakthroughs, there was one particular one that I think has really shaped our understanding of stem cells, but also of cell identity as such. And that was the discovery of Shinya Yamanaka, who showed that you can turn probably any cell back into a stem cell by activating a code of transcription factors. 4 different transcription factors. If you activate them together in a cell, they can turn a cell back into a stem cell state. And of course, that's mind-blowing because it allows you now to create stem cells from any individual. It took away all the ethical constraints of working with human stem cells. And it also means that every one of us has their own repair kit, in theory. Now, This inspired a colleague of mine called Marius Wernick in Stanford, who thought maybe that concept of using transcription factors to program cell identity could be generalized. And what he did was he developed a protocol that allows you to turn a skin cell directly into a brain cell. And then another program that allows you to turn a stem cell directly into a brain cell, and ultimately even a liver cell directly into a neuron. Not quite sure what this is useful for, but it serves to show the point that we have to rethink the concepts of cell identity. What makes a cell a cell? And it suggests that it's radically different from what we used to think, that cells are defined by their history. It's more likely that cells are actually the identity of a cell is contained in the cellular state at the moment, that is driven by transcription factors and gene regulatory networks, so programs.
That's a pretty, I guess, profound distinction. I'd love to hear more about that. So if that's true, where cellular identity is about transcriptional state and there isn't a path dependence, or maybe not as much of a path dependence as we might think, what does that allow us to do that maybe wouldn't be possible in the previous paradigm, where path dependence and the past history of a cell had some fundamental impact on its future?
Well, I think it changes everything. If the identity, the sub-identity, the state of a cell is defined by the active genetic programs and not by the history where the cell accumulates epigenetic changes in order to then form a particular cell type, we can completely rethink how we generate cells, how we manufacture cells, and what might be possible. So one of the interesting origins of all of this research is really a string of scientific investigations and papers that were conducted in the 1980s, when Weintraub and Lazar essentially demonstrated that you can turn a skin cell into a muscle cell. And that was the very first transcriptional reprogramming protocol. And he started these speculations. And one of the questions that he asked is whether it might be possible to create hybrid cell types, cells that have traits of two completely distinct cells, like a pigment cell, a melanocyte, and a muscle cell. And he showed that this is possible, which essentially means that in this paradigm, you can not only create cells that exist in the body, but you might be also able to create cells that actually don't exist within the body, but that are possible from the genetic network point of view.
That's fascinating. So from a rejuvenation therapy standpoint, or in a medical application, I'd love if you could paint the picture of what this opens up from here and where do we go from here. And I'm also really interested in digging into whether you see a future where individual persons, my cells, are taken out of my body, reprogrammed, and then put back in me to treat my kidney disease or brain injury or whatever it may be in the future. Or is there a world where it's more of a, to use a more technical term, allogeneic stem cell therapy, where there's a bank of universal donor type cells or large subset of cells that could be transplanted off the shelf, so to speak, into people? Where do you see the future going, and what are the hurdles that we've got to face?
Well, a lot of questions. So the first one is the way of rejuvenating people. And I think, yes, there is also something like the aging program, and it's also probably driven by a transcriptional state. But I think that sort of leads us down a completely different aspect. What can we do with cells that we can produce from pluripotent stem cells? We can use them as intelligent medicines. So what makes that medicine intelligent? The cells can replace cells that are lost. That's, of course, unique. You can't have that with small molecules or biologics. And the other thing that's unique, the cells can adapt and respond to their environment. And that's probably the reason why we've seen this incredible breakthrough with the first cell therapies that entered our clinical setting. CAR-Ts are, I would say, the brother right flight of cell therapies. These are T cells that have been engineered to identify tumor cells and wipe them out. And they've shown to be incredibly powerful in the context of blood cancers. So patients at the end of treatment can now receive a cell therapy that essentially eradicates the tumor. And that's incredible. This particular early stage, very expensive treatment is autologous, i.e. these are cells derived from the patient and engineered. To me, that is more an experimental medicine paradigm. Why? Because at the moment, the cost of this is incredible. In the UK, I think the NHS pays more than £350,000 per treatment. So this is certainly not a treatment that can be rolled out to everyone, and certainly not in lower income countries. And for me, the definition of a medicine is that it needs to be available to everyone. So we need to push beyond this personalized approach at this point in time. And there are really two opportunities here, as you mentioned. And I'm not quite sure which one is going to win. Probably they're going to sit next to each other, side by side. One is the autologous approach, where you take a cell from an individual, turn it back into a stem cell, and then create the cell type that you require for your treatment. For example, a dopaminergic neuron if you suffer from Parkinson, if you suffer from diabetes, a pancreatic islet cell. And by the way, these have been shown to be efficacious in a clinical setting. But in order for this to become a medicine, by my definition, i.e. available, we need to really commoditize the process by which cells are turned back into stem cells and then turned into the cell type that you want. And that's going to be extremely difficult. It's going to happen over maybe a decade or so. But at this point in time, this approach is extremely costly. So the alternative approach then is to somehow try and match a bank of cells to the patient that requires the cells, the allogeneic approach. And here, there's really three options. There's a number of cells that it turns out you don't have to match. So, for example, if you want to treat an individual with an NK cell that you again program so they recognize a cancer, it has been demonstrated that the NK cell doesn't need to be matched. So you can take anyone's NK cell and transplant it into the patient. And that, of course, makes for a huge reduction in cost for this approach. Not many cells can be transplanted without having to match. But we actually know how to match cells. If you think about it, we've been doing organ transplant now for decades. So we're very versed with the paradigm that allows us to match organs with patients. And we can do the same with cell therapies. And the great thing about cell therapies is that you can actually tap into what I would call super donors. So these are individuals that have the same HLA alleles on both chromosomes. And with a very few of those, you can match a wide population of individuals. I'll give you a number. About 3 of these super donors, if you pick the right ones, can be matched to 50-60% of the population in Europe, or 12 of them can match 99% of the population in Japan. So that means you can build a relatively small bank and still provide cells off the shelf. So the cost reduction here, I imagine, is about a factor of 100 to patients. And then, of course, the third approach that you mentioned is maybe there's a way to make cells universal. Maybe we can create strategies for them to evade the immune system. And that has been achieved by quite a few labs so far. Obviously, you have to edit surface molecules, etc., in order to achieve that. But if that turns out to be successful in human beings, and if it turns out to be safe, then that will give you another option of really reducing the cost rapidly, but also making sure that the cells that are produced are of a much higher quality and much more specific than if you'd say you take primary autologous cells, which are always a mix, and you never know what you're getting.
You at bit.bio, you and the team have attracted a lot of interest from a lot of very smart scientists, very smart venture capitalists and funders to help you to build this vision. I'd love to hear more about what it is that you all are doing really differently and how you're tackling some of these challenges that you've outlined.
So bit.bio is really built on this paradigm of cell reprogramming, which basically means we can now, with our technology, turn a stem cell into probably any cell type within days, and we can get pure cultures. That is something that everyone dreamed, but nobody thought possible. And and the reason why we can do this is because we have understood the biology behind cell reprogramming, a process called gene silencing. A cell essentially recognizes if you try and switch on a different program and tries to shut it down. So we had to trick them into accepting the new program. And we did this using an approach that targets genetic safe harbors. We call it opti-ox. And it optimizes the inducible expression of transcription factors or other genes. And so this creates a platform by which we can turn the fuzzy logic of biology into something that is deterministic. And it's extremely scalable as well, by the way. So we have a sister company, Meatable, that just announced the first product, a sausage made of porcine iPSCs with fat and muscle cells. And they're now in the trillion cell range on a weekly basis, pushing beyond now that the kilogram range. Obviously they have to go into the ton and then kiloton range, but it is possible. This biology scales incredibly. And so that's one pillar. I would say that's our manufacturing platform. The other pillar that we had to build is what I call the discovery platform. So because nearly all stem cell research focuses on directed differentiation, which is essentially using chemical cues in order to recapitulate what happens during development. The space of transcription factor reprogramming, the knowledge space, is very patchy. So very little is known. So there are not many cell programs out there. And what we wanted to do is we wanted to be in a position where the company can create any cell type from scratch. So let's say tomorrow morning I wake up and I say, I'd like to have a pancreatic islet cell. We can do that. We have built the systems that allow us to screen transcription factor combinations in very high throughput and scale. And we've systematized the discovery process and then the translation into product. So the productization process so that we can actually produce this. I think this is totally unique. I don't think there's another company or even lab that can do that. Essentially create cell types de novo. The competitors that we have, they're mainly based on in-licensing of existing IP. So we can create, in theory, every human cell type, and then we can manufacture it as well. And so this is this is huge if you think about the opportunities. And that's a blessing, but also a curse. Why is it huge? Every cell in the body is a potential cure, is a therapy. So that's the potential. It's also a thing that you can use to do research and drug discovery. So these are the applications. Again, a very sort of important, impactful opportunity. But as a startup, of course, you have to, and although we are a late stage startup, so we've raised about 200 million so far, we have to be careful with our focus. And so we have to show the depth of our platform, in this case, how clinically relevant the platform is, in addition to the breadth of the platform, demonstrating that we can actually do what we say. We can actually add cell types ad libitum. So that's where we are at this point in time. This transition from a sort of a late stage, I would say, startup, scale up stage to a company that hopefully in the not too distant future is in the clinic, demonstrating what this technology can actually do.
Yeah, I think that's perfect. And you talked about this breadth versus depth challenge. I'd love to hear more about where you all are going deep and where the field is going deep. What are the types of diseases or cell types that are going to have the biggest impact first?
So I can't announce what we're doing right now, but if you think about it theoretically, you want to find a application of human cells that is de-risked as much as possible, i.e. where there has been efficacy or there has been a very strong clinical signal in a previous clinical trial. So that's one criteria, I would say. The second criteria is you want to have a condition where you don't have to think about or too much about the matching question for the first pass into the clinic. You just want to make sure that the cells do what they need to do in order to help a particular indication. So we're obviously talking about an autologous approach there. And there's ways of making cells autologous. Encapsulation is another thing that I haven't actually discussed yet. That's the route that Vertex has taken, for example, with their pancreatic islet cells. And another paradigm perhaps is think about cells that have large impact independent where you need to put them. Administering cells into the brain is a much higher barrier than, say, infusing them into the bloodstream. So those are the criteria by which I would say you can decide what is a good first path into the clinic.
That's really helpful, and in many ways more helpful than naming any specific diseases, because I was going to follow up and ask about the framework and actually how you think about these. Is it fair to say that delivery is one of the big challenges across the board? How do you get cells or cell therapies to the right tissue? Because you mentioned the challenge of the brain compared to blood or liver and others. That seems to be a challenge that almost everybody in the cell and gene therapy space that we talk to comes up against in one way or another.
It varies, as I said. Infusing something into someone's blood is typically pretty easy. Getting the cells to the patient might be tricky. So the logistics of it, especially if you take autologous cells. The ideal version here, of course, is if you've got an off-the-shelf product that is frozen, that you could just take out of the fridge essentially and administer. And that's really the holy grail. Obviously, that's something that we'd like to deliver. But there are organs that are very delicate, very, very, very difficult to reach, where cells have to be administered in a very particular location, or where they have to integrate in a very particular way. So let's think about the brain again. In most cases, even if you've got the most beautiful neurons, so it won't suffice to put them into the brain, because they would then have to create connections with other neurons, and they have to sort of, some of those connections will be very distant. So they'd have to navigate their processes across a very complex organ. And I think that is going to be a challenge all the way. Other cell types in the brain might be easier. So if you think about the glial compartment, for example. But the good news is that in some instances, you can actually choose a location for the cells that isn't the original location. So let's go back to this wonderful example of the pancreatic islet cells. It was like, for me, that was like an earthquake when they announced the data. The first patient that was treated with a pancreatic islet cell was cured from his diabetes, did no longer require insulin. And they did not put the cells into the pancreas. They were injected somewhere entirely different, somewhere where you can, with much easier access. So there's that option as well.
Have you all, and I'm sure you have, thought about this, comes down to the focus question, but it seems like your platform could also help to answer a set of really interesting questions around exceptional people. And what I mean by that are the people who have ApoE4 but don't get Alzheimer's, or the people who have C9 or 7-2 but don't go on to develop FTD or ALS. And there may be something in their genome or otherwise that is protective. How do you think about some of those types of applications where you maybe create a bank of cells or a single cell line from people like that might be exceptional? And we could think about all sorts of interesting, exceptional examples. I'm sure your team comes up with new ones every week.
Well, now we're talking obviously about the use case of these cells as a research tool.
As a research tool, yeah.
And I mean, yes, you can do incredible things with this approach. So just to summarize, what we can do and what we've shown we can do is create pure cultures of individual cell types that are highly specified. So not only cell type or cell identity, but also subcell identity. So we can, for example, create a dendritic type 1 versus 2 cell, or a particular form of a sensory neuron versus another sensory neuron. And what you can then do is you can combine very precisely these cells with each other to create more complex model systems. So your question is, why are certain mutations not penetrating into disease phenotype? And that's a huge question. It could be, of course, that mutation requires a particular genetic background that has certain risk genes that enables that. It could also be epigenetic, of course. And it's an incredible question. It's something that we have seen when we induced and created disease models. Cells that have exactly the same genetic edit, but don't display a phenotype. Right. In my academic lab, I had a family that we were able to study using stem cells, where the parent had the same mutation as the son, and the parent there was no phenotype, and the son was heavily autistic. So you can see exactly that question, and the tools now are here to pick these apart. I think that's extremely interesting. And the other thing that I find extremely interesting is you can unpick the disease pathology in the context of a more complex tissue. So what you can do is, for example, you can mix, let's say, brain cells that have a disease mutation, neurons, let's say, with microglia that don't have the disease mutation, and see whether having the mutation only in one cell population is sufficient to drive the phenotype. And vice versa. You can, of course, then create diseased microglia and wild type neurons, just as an example. So it's an incredible way of unpicking human disease, which is very different from, in a way, from the animal models, because they are always in a different species, and that has massive limitations.
We're coming close to the end of time here, and I wanted to just ask you, maybe on a more personal level, how you found the shift from practicing medicine to, I think you were hybrid practicing medicine and doing research at the same time, to now, I suspect you're not seeing patients anymore. You're spending most of the time building the team and running the company. How has that been? How's it been for you? What's better than you expected? What's harder than what you expected?
I'm still actually seeing patients. Yeah. But so what have I learned? So one of the big lessons right at the beginning that I got awfully wrong, and then fixed with huge focus and intensity, was, in a company, the team is everything. In fact, a company is the team. And if you make compromises here, you are damaging the company. So you've got to make sure that the people are motivated, aligned, and that you can work with them and they can work with each other. So that is something I learned the hard way. And it's been the reason why we then implemented a very, very intense, let's say, screening or recruitment process. And we defined a core set of values that people have, that we're very open about communicating with anyone. so that people can make their own choice as to whether they fit into bit.bio or not. And we also try and live these values. Nothing is perfect, obviously, but we really try to embed them into the organization. And this has allowed us to create something that I think is very special. So bit.bio has a very, very strong culture that I think any visitor will sense the moment they come to us.
What do you think is the most unique aspect of the culture? One or two?
So if you think about it, this is a very deep scientific problem that we have to crack. So it requires a lot of incredibly talented scientists, but not only that, it requires incredible engineering, incredible manufacturing, people that are interested in doing something that's never been done before. So the act of creation is really important. But often you see that the more brainy someone is, the less good it is at communication. And so we had to find a way of picking individuals that make the effort to connect and to come together. So what are the 4 values? Very briefly, the first one is purpose. bit.bio's purpose is to program cells to enable new paradigm in medicine, novel cures, and novel cell therapies. So we're very open with that. Now, people that are thinking of coming into the company can see whether their own purpose matches with this purpose. And it doesn't have to be the same, by the way, but you could be a chap who likes finance, but wants to do something good rather than impactful, rather than, perhaps, running a hedge fund or something like that. It could be a scientist that is deeply curious about cell identity, or it could be someone who's really, really passionate about translating something into the clinic. So it provides an umbrella. And of course, when your purpose matches the company's purpose, you immediately are motivated. What we're trying not to stay away from is people that don't know. I don't know what my purpose is in life. Those will not, feel very, very home in bit.bio. The second is ambition. There's, of course, two sets of ambition. Ambition towards the purpose, that's what we're looking for, or self-optimization. That's something that we're very, very negative against. So this is part of the screening process. The third would be then collaboration and trust. Collaboration only requires trust. Again, there's 2 ways of trust. There's, I would say, the lazy trust. We've known each other for the last 10 years. You've always been on my side. I trust you. And there's this other trust, which is leaning forward. I don't know you, but I trust you. And that's an effort. So we're looking for people that can make that effort. And finally, we're a science company, so we need to be empirical. Facts above opinions. So that's the core 4, I would say, that we've formulated. And that has allowed us to really create a very unique team, very strong, very communicative, but at the same time, very deep scientifically. I mean, I'm a nerd. I think we're a company of nerds, but I would say this is the badge of honor.
Yeah, and I've interacted with a number of members of your team, and I think they live all of those values that you just described, and I think what you described as well about having not just smart scientists, but smart scientists and great communicators is absolutely true and critical. It makes it harder to find people, but you've got to be patient and rigorous, right?
Yeah, I'm glad you say that. Yeah, no, absolutely. It should be difficult to join bit.bio and easy to leave it.
Yeah. Well, Mark, I know we're up against time. Thank you so much. I think this was I appreciate you entertaining all of my left field questions about where this may be going and where it is today. Is there, if people want to find you, they can visit you on your website, bit.bio.com. I assume you're always hiring, the rate at which you're growing. And for you personally, you're on, they can find your team's contact info on the website. Are you on social media?
Not enough, I'd say, but I need to up my game here. But thank you for the conversation. And I really enjoyed it.
Great. And thanks everyone for listening. And as always, please share with a friend if you liked the episode. And we'd love if you could leave us a review on your favorite podcast player to help other people find us. Thanks for listening and see you next time.